In aim 3, we will investigate if there is any modification in the host dendritic cells which lead to the direct interaction of Bifidobacterium longum with the host cells. Pattern recognition receptors are a key element of the innate immune system that is equipped to detect pathogens such as viruses and bacteria. They are mainly expressed by antigen-presenting cells such as dendritic cells and macrophages, but they are also found in other immune and non-immune cells. The PRRs are divided into four families. Two among them that are widely studied are toll-like receptors and nod-like receptors. The toll-like receptors are transmembrane proteins that recognize the conserved molecular structure of microorganisms. The nod-like receptors are cytoplasmic proteins that regulate inflammatory and apoptotic responses. Tumor-infiltrating dendritic cells are mature active DCs that have infiltrated into the tumor to increase immune activation and produce anti-tumor immune response. A recent study showed that the allogenic stimulatory capacity of tumor-infiltrating dendritic cells differed from that of the DCs in lymph nodes. They also see changes in the characteristics of TIDC over tumor progression. Here, they see the expression of IFN gamma, granzyme B, and perforin. Granzyme B is a serine protease produced by cytotoxic lymphocytes and natural killer cells, along with pore-forming protein perforin to mediate apoptosis in target proteins. So they see an increase in the production of these proteins in late tumor-infiltrating dendritic cells compared to the early tumor-infiltrating dendritic cells. Beta is a non-DNA binding coactivator that serves as a master control factor for MHC2 expression. IFN gamma is known to activate zeta to produce MHC2 molecules. We are interested in zeta because a previous study showed that when Bifidobacterium is fed to tumor-bearing mice, there is increase in the expression of MHC2 molecules in the dendritic cells in the tumors. Their data pointed to an improvement in immune response of the Bifidobacterium-fed mice upstream of T cells at the level of host dendritic cells. Our hypothesis is that there is modification in the pattern recognition receptors of the tumor-infiltrating dendritic cells, especially the toll-like receptors and nod-like receptors. This enables them to detect and interact with the Bifidobacterium longum, which then modulates the zeta gene by increasing the production of IFN gamma, and hence increasing the MHC2 molecules, which then recruits other immune cells. In order to test our hypothesis, we'll perform comparative analysis of the gene sequence of the toll-like receptors and nod-like receptors of TIDC, tumor-infiltrating dendritic cells, and compare it with that of DCs from the lymph nodes. They are the controls. We will also investigate the expression levels of the TLR and NLR in the TIDC to understand the gene activation profile of TLR and NLR and their downstream proteins. We will quantify the steady-state mRNA levels by RT-PCR. To pinpoint the key players in the immune response, we will, based on the previous experiment model,